This is the audio version of ICH Q7 table of contents. Reading as it is given in the table of contents of ICH Q7. There are 20 sections and there are some subsections. We are reading all given as it is. Table of contents. 1. Introduction. 1.1 Objective. 1.2 Regulatory applicability. 1.3 Scope. 2. Quality management. 2.1 Principles. 2.2 Responsibilities of quality units. 2.3 Responsibilities for production activities. 2.4 Internal audits, self-inspection. 2.5 Product quality review. 3 Personnel. 3.1 Personal qualifications. 3.2 Personal hygiene. 3.3 Consultants. 4 Buildings and facilities. 4.1 Design and construction. 4.2 Utilities. 4.3 Water. 4.4 Containment. 4.5 Lighting. 4.6 Sewage and refuse. 4.7 Sanitation and maintenance. 5 Process equipment. 5.1 Design and construction. 5.2 Equipment maintenance and cleaning. 5.3 Calibration. 5.4 Computerized systems. 6 Documentation and records. 6.1 Documentation system and the specifications. 6.2 Equipment cleaning and use record. 6.3 6.3 Records of raw materials, intermediates, API labeling and packaging materials. 6.4 Master production instructions, master production and control records. 6.5 Batch production records, batch production and control records. 6.6 Laboratory control records. 6.7 Batch production record review. 7 Material management. 7.1 General controls. 7.2 Receipt and quarantine. 7.3 Sampling and testing of incoming production materials. 7.4 Storage. 7.5 Re-evaluation. 8 Production and in-process controls. Production operations. 8.1 Production operations. 8.2 Time limits. 8.3 In-process sampling and controls. 8.4 Blending batches of intermediates or APIs. 8.5 Contamination control. 9 Packaging and identification labeling of APIs and intermediates. 9.1 General. 9.2 Packaging materials. 9.3 Label issuance and control. 9.4 Packaging and labeling operations. Storage and distribution. 10 Storage and distribution. 10.1 Warehousing procedures. Warehousing procedures. 10.2 Distribution procedures. 11 Laboratory controls. 11.1 General controls. 11.2 Testing of intermediates and APIs. 11.3 Validation of analytical procedures. See section 12. 11.4 Certificates of analysis. 11.5 Stability monitoring of APIs. 11.6 Expiry and retest dating. 11.7 Reserve or retention samples. 12 Validation. 12.1 Validation policy. 12.2 Validation documentation. 12.3 Qualification. 12.4 Approaches to process validation. 12.5 Process validation program. 12.6 Periodic review of validated systems. 12.7 Cleaning validation. 12.8 Validation of analytical methods. Section 13 Change control. Section 14 Rejection and reuse of materials. 14.1 Subsections. 14.1 Rejection. 14.2 Reprocessing. 14.3 Reworking. 14.4 Recovery of materials and solvents. 14.5 Returns. 15 Compliance and recalls. 16 Contract manufacturers, including laboratories. 17 Agents, brokers, traders, distributors, repackagers, repackers, and relabelers. Applicability. 17.1 Applicability. 17.2 Traceability of distributed APIs and intermediates. 7.3 Quality management. 17.4 Repackaging, relabeling, and holding of APIs and intermediates. 17.5 Stability. 17.6 Transfer of information. 17.7 Handling of complaints and recalls. 17.8 Handling of returns. 18 Specific guidance for APIs manufactured by cell culture or fermentation. 18.1 General. 18.2 Cell bank maintenance and record keeping. 18.3 Cell culture fermentation. 18.4 Harvesting, isolation and purification. 18.5 Viral removal, inactivation steps. Section 19 APIs for use in clinical trials. 19.1 General. 19.2 Quality. 19.3 Equipment and facilities. 19.4 Control of raw materials. 19.5 Production. 19.6 Validation. 19.7 Changes. 19.8 Laboratory controls. 19.9 Documentation. 20 Glossaries.